So just the lymphatic vessels from this lecture will be on our upcoming exam on Thursday. You guys also realize that you can take your exam on Thursday evening or Friday at noon. So either time's fine. Just remember, hour and 15 versus 50 minutes for the exam. So keep that in mind. You're welcome to either one. So lymphatic system. This is going to be the system that's going to take that excess fluid that's left in the interstitial spaces after we get through the capillary bed and bring it back to our circulation. The lymphatic vessels should be thought of as drainage or overflow vessels. They are interwoven in with our capillaries. So we have lymphatic capillaries that interweave within our regular capillary bed, and that allows them to therefore be there in the area where the overflow fluid is being left. And our notes say lymphatic capillaries are interesting because they are blind-ended. So our regular capillary bed goes from an arterial side to the capillary bed to then the venule side. Well, with our lymphatic capillaries, the only start is the very beginning of the capillaries. There's nothing that they are actually in between other than they're sitting in that interstitial fluid. So we have our picture here, which is showing the arterial side and then the venule side and the capillary bed in between. And now interwoven are these blind-ended tubes that are there to take care of the overflow of the interstitial fluid. And the reason they can do this, they can take this interstitial fluid which is accumulating in there, is because of the construction of our lymphatic capillaries are very different from any of our other capillaries that we've encountered. They do not have tight junctions, so no tight junctions connecting the endothelial cells. Instead, the cells, instead of coming together, have an overlapping aspect to them. And this overlapping creates these mini valves, or definitely I call them pseudo valves. And attached to the flappy portion of that endothelial cell that's overlapping are these connective tissue filaments. So they are going to anchor that flap that's overlapping to surrounding cells. So they'll have a little line, a CT thread going to them, and when we get swelling, we're going to get increased fluid in that space, and it's going to pull on that little thread, and it's going to open up that flap, and that's going to be what allows the lymph to actually enter into that area. So increasing interstitial fluid is going to cause swelling or edema, and this is going to cause the CT to pull open the valves, and I'm putting valves in quotation marks. So this will allow all the kind of fluid that's in the interstitial fluid, all the stuff that's in it, to actually enter freely. So it's unrestricted entry into this. And so it doesn't matter how small or how big you are. Fluid's really small, flows on in nicely, but we can have protein, we can have cellular debris, we can have microorganisms. All those kinds of things can actually enter into the lymphatic capillaries if they are present. But if it's just regular interstitial fluid, what should the color of lymph be if it's just regular interstitial fluid? Clear. It indeed is clear. So lymph is clear. But when you have cellular debris and microorganisms, what does that tell you is going on in the soft tissues? It's an infection. And so don't you want that material out of there? Isn't it kind of nice that we have this little drainage system that will also help to drain those infectious agents and debris that's actually building up in those areas to get them out? Because also remember, the more stuff that you have in the interstitial fluid, the more increase in osmotic pressure you have, which will then, of course, make the swelling even worse, which, of course, will help flush the things out. You guys follow that process? So it's a really nice process for us. Now, we're going to quickly do a little bit of a blood and lymph capillary diagram. And so I'm putting this tube here in the middle for blood, and I'm making my endothelial cells on it. It's easiest to do it this way. Nuclei. So this is a capillary going through a tissue bed that I'm going to draw. And remember, we're capillary dynamics. We deal with hydrostatic and osmotic pressures. And on the arterial end and the venous end, so A versus V, remember we have a net capillary hydrostatic pressure of 35 minus our net osmotic pressure of 20. So that gives us, yes, that gives us our 15, which means fluid leaves, correct? On the venial end, let's do CHP on the venial end is 15. Our net osmotic pressure is 20 minus 20, so that gives us that number of a -5, which means, so we have 15 out, and a -5 means push fluid out or pull fluid in? Pull fluid in. So we're going to have 5 in, and that's what the negative actually refers to, because it should be -15, shouldn't it? So what we have here, the differential is then between these two is 10 millimeters of mercury worth of fluid that's trapped in the interstitial spaces. Well, what we have on the other side, I'm going to draw it on the other side because it's easier because I have my numbers on one side. I'm just drawing cells of the interstitial spaces. And so these cells have also those lymphatic capillaries interwoven in there, and so I'm going to do a little bit of a weave here. And I'm going to finish my capillary, and I'm going to make it like this. I'm going to overlap. Let's put our little nucleus of our endothelial cell so we remember. And same thing on the other side, so let's do some overlapping. Our picture's not the best, but I hope that the message is getting across. And so what happens here is that we have CT attached to connective tissues, threads attached to the nearby overlapping exterior part of that endothelial cell. So as we get increasing fluid, which is what that number here up top means, increasing 10 units of fluid, then we are going to start pulling on that flap, which is then going to allow the fluid entry into that lymphatic capillary, and it will start to go out of the tissues. So understanding capillary dynamics, what leaves on the arterial end and what comes back on the venous end is important, because that is the fluid that is bathing our tissues, giving it the nutrients and oxygen and hormones that those tissues need. And then on the other side, we're pulling that fluid back in, which is considered waste material. You know, oxygen poor, carbon dioxide rich, nutrient poor, waste rich blood that's going to go back to the various organs that are going to be responsible for giving us more oxygen, getting rid of carbon dioxide, giving us more hormones, getting rid of the wastes, etc.
Is it still hydrostatic pressure on the venial end that you have there? Or is that?
Capillary hydrostatic pressure? Yes.
Still hydropathic, it's not
Yes.
osmotic.
Yeah. This one here is the osmotic.
Okay.
So it's our net pressures have to take into account our osmotic pressures. So we have hydrostatic pushing the fluid out, osmotic, pulling the fluid in, and so whatever amount of fluid leaves or enters is due to that balance. Let's take a look at the organization of the lymphatic vessels. Okay, lymphatic capillaries first, the smallest. Then the next larger size are called literally lymphatic vessels. They indeed have the same tunics as veins. And how many do veins have? 3, so they still have the 3. And they have valves like veins. Now the venules don't have valves, but the veins have valves. And so what's really interesting, these are really small vessels, because if we just got out of the capillary, if we were to compare it to an artery side or a venule side, we would say these are about the size of arterioles or venules. So they have the same tunics as we discussed earlier with our blood vessel anatomy, but what is important is these guys, starting at the next larger structure, at the lymphatic vessels, have a lot of valves in them. And this is really, really important in getting our lymph back, because the valves are just like the valves we talked about for the venous system. They are designed to help that blood get back towards the heart. So they're one-way valves, just like the semilunars. So capillaries, lymphatic vessels, then we're getting larger, and we now come to what are called lymphatic trunks. And these are indeed formed by the union of the larger vessels and are visible. You guys can actually find them. And when we find the trunks, they're typically associated in various areas. And we'll give them names, because now we can see them. They are large enough to actually give them names to. So we have jugular trunks. We have intestinal trunks. We have lumbar trunks. We even have mediastinal trunks. So those names tell you where to find them, where they are bringing lymph from. So capillaries, vessels, trunks, and now ducts. That's the final size that we will actually find them in. And there are only 2 ducts, and our notes state largest of the lymphatic vessels, and both of them, so there's just 2, there's a right and the left, both of them are going to enter into, they are going to take the lymph that they are carrying into the venous side of our circulation, and they are going to dump it into the junction of our internal jugulars, which is a vessel in our neck, and our subclavian veins. So 2 sides of the neck, so we have 2 internal jugulars, and we have 2 subclavians, and right where they actually join, that's where those are going to actually, those ducts are going to empty into those areas. So right in the little V of those spots. Now, it's kind of weird, but this is just the way it is. The right lymphatic duct drains only the right side of the head and neck area and the right upper extremity. That's it. And so what we have over here, this is the right side of this person. This is the left side of this person. And so what we see right here, this is our internal jugulars right here. And here's our subclavians right here. And so the right lymphatic duct is coming in right there at that junction, but it is only draining the right side of the head and neck and the right upper extremity. So it's really short, and it's small or attenuated. It's difficult to dissect out, but you can figure that the jugular trunk of that duct is going to be laying alongside the internal jugular vein. Now, the main duct is called the thoracic duct because it's easy to find it in the thorax. That's right. That's why it's called thoracic duct. And also the left lymphatic duct, and it drains the rest of our body. So what that means is it is bringing lymph from our lower extremities, both lower extremities, from the abdominal pelvic area and from everything in the thorax, as well as the left upper extremity, left head and neck area. So it drains everything else. It's just the way it is. It's an odd pattern, but it is what it is.
Do they know why?
No.
I have no idea.
No idea. This is just the way it evolved. And it's in cats, dogs, humans. I don't know about other critters, but this is our system. There is an interesting collection of lymph below the diaphragm called the cisterna chyli. Cisterna chyli, and that is this area right there. That's that bulging area, and it is receiving lymph from everything below it. So lymph from our extremities and the abdominal pelvic area and everything is going to empty into the cisterna chyli. And a cistern is a collection of water. So this is an area where all the lymph is collecting from areas below it. And then, like a lake, there's a bigger river that leaves the lake. So the trunks empty into the cisterna chyli, and then the duct, the thoracic duct, leaves that area, follows meanders along the aorta in the thorax, and is actually dorsal to the aorta. So when I say dorsal to the aorta, that means it's on the backside of the aorta. So that's one of the things that's really interesting. It's laying behind a vessel that's an elastic vessel that is always springing on it. In other words, jumping on that duct. And remember, it has valves. So you can figure out that's actually a good idea, that you are in this free cavity, and in order to squeeze your lymph back, you want somebody squeezing you. And so it's lying on the backside of the aorta, and as the aorta bulges, when we get our pulse pressure going through that and everything, that aorta is going to milk that duct, and is going to help the lymph actually come back to the internal jugular and subclavian junction on that side. And so we see, as we follow this thoracic duct up, it has a tendency to get more superiorly turned to the left side, and then goes behind the subclavian. I hope you can appreciate this. It goes behind the subclavian, internal jugular, and then joins on in at that junction area. Now, in addition, when it makes that nice hairpin turn, what we're going to have is other feeders, other trunks coming into that from the left head and neck and left arm area, upper extremity. Got it? All right. And that brings us to lymph transport. Unlike blood transport is pumpless. In other words, remember the real pump for blood is the heart. And it's interesting, we talked about their construction. They have the same three tunics, and our notes here talk about rhythmical contraction of lymph vessel walls. So that's saying that the smooth muscle is trying to milk the lymph back, and this is very weak. So that's not the easiest way to get our lymph back. The easiest way to get our lymph back is the same factors that got venous return back to the heart. And so when we say muscular pump, remember that's not a real pump. We're talking about what muscles? Skeletal muscles playing here. And then ventilation, or the respiratory pump, we're talking about pressure changes in the thorax, helping lymph to get back into the venous side of the system. And then the big player for lymph is that they are just super rich with valves. And sometimes in some specimens that when we've done some cat dissections in the past, which were different from the cats that we have, you could literally see beating all along the duct. So it was really interesting when you had somebody that had a lot of lymph in their lymphatics. If they are, what do you suppose? Will you have more lymph or less lymph in your lymphatics if you're well versus sick? Which one? More if you're well or more if you're sick? More if you're sick. Isn't that correct? Yeah. Isn't that one of the things, too, that you have to realize that we're going to talk about the next thing is what's attached to our lymphatics that is also a very important feature to hang on to them, because we just talked about if you have an infection in an area, that will increase fluid and edema in that area, correct? And debris and stuff like that. And so that's going to enter the lymphatics. And don't you want to get the bad stuff filtered out before it gets into the bulk of your circulatory system? So If you are ill, you'll have more lymphatic activity than if you are well. Lymph nodes, there are hundreds distributed throughout the body, but we have them actually concentrated in our cervical, axillary, and inguinal areas. But there are interwoven throughout. It looks a bit like a spiderweb pattern or a lacy pattern there. And so things start distally. And what's nice here too is it shows that construction of the lymphatic capillary with the pseudovalves, but actually in the real lymphatic vessels, we have all these lovely valves that are allowing us to get the lymph back in the proper direction. Now, lymph nodes, they function to filter lymph. Speller's off. Fine. So if there's no infection, the lymph goes through really quickly. But if there is an infection, then we're going to actually slow down the filtration so that we can actually pull more stuff out. Our lymph nodes are major sites of immune system activation. So let's take a look at lymph nodes now. Lymph nodes have a distinct structure to them. They have a capsule, as most organs do. It is dense, fibrous connective tissue, but what's really interesting, it is actually loosely attached. Most of our capsules are tightly attached to the organs that they are covering up. Inside our lymph node, we are going to find a lot of reticular fibers. You guys remember reticular fibers are made out of collagen, but they're thin and lacy-like. So they have a reticulated or interwoven pattern. They hold the stroma, and stroma is a term that is referred to as the functional tissue of the lymph node together. And we're going to find reticular fibers in all of our lymphatic organs except the thymus. The thymus doesn't have reticular fibers in it. It's the exception to the rule. Cortex always means outer region, and so then the inner region is the medullary area. Let's go ahead and move on down here. And we can see, let's give this guy, this would be the medullary area, and I've got a little bit of cortex in there, but for the most part, it's going to be kind of a little bit of an area where everything comes together for the final vessel to leave, and the vessel to leave is going to be emerging from an area of the lymph node. In your mind, your lymph node should be shaped a bit like a bean. That's called the hilum, or hilus if we're talking about more than one, and the vessel leaving it is an efferent lymphatic vessel. We've used efferent and afferent before, so this is leaving the lymph node, and so those entering the lymph node are called afferent lymphatic vessels. One of the things that's important is to note already how many afferents do we have to efferents. We have a lot of afferents, very few efferents. So what does that mean if a whole bunch of lymph is coming to that lymph node? It's going to slow down big time, isn't it? So it's going to slow down in the lymph node, and that will allow for proper filtration to occur and activation of the immune system, especially if there is a lot of lymph associated with any sort of infectious process. So this construction is very useful for filtering out the bad stuff. The other thing that's very important is in the cortex, there are these nodules. And in the middle of the nodule is what's called the germinal center. Germinal means what to you, or germinate means? To grow, right? To plant, to grow. And so This is very important because in the germinal centers are dividing lymphocytes, and they're a very specific type of lymphocytes. They're B lymphocytes. I'll write that down in the notes in just a moment, but that's very important. Germinal center, hot zone for dividing B lymphocytes. So those are part of lymphatic nodules that we also call follicles. So they will often go by the same name. Let's take it's a synonym or analogy. Let's take a look here in this picture. That's a lymphatic nodule or follicle, and it's showing you down below. The germinal center is in the middle of it, and it is lighter, and there is a darker mantle around that germinal center. The germinal center, again, is for B lymphocytes, and the area around it is for T lymphocytes. Medullary cords are obviously in the medullary area, and trabecula, remember trabeculae carneae? Those are also cords of CT that actually invaginate or go into kind of the meat of that lymph node. Let's go ahead and go back to the black ink. And in the cortex, in the outer region, let's add some notes off to the side here. Let's make sure that we state that's where we find densely packed spherical collections of lymphocytes called follicles, and sometimes nodules, depending upon the literature, containing a center that stains lighter and is called germinal center. And here's where we're going to find B cells. B cells are the same thing as B lymphocyte cells. So if we're going to draw our own follicle, you have a darker outer aspect, and here are our B cells, and the follicle will have a lot of T cells and macrophages on the periphery. Going down, our notes say B lymphocytes predominate in the germinal centers. That's not, so in other words, they are in other areas, but that's where you are for sure going to be able to find them and identify them. T lymphocytes are found in the rest of the cortex. And then there's these interesting new cells. We've actually heard about them before when we talked about the skin dendritic cells. That's way back to last semester, a little bit of skin topic. And these guys are very abundant in the cortex, and they are special macrophages. And they will interact with the immune system. And we'll get into that immune system, which are our T and B cells, T and B cell system. When they interact, they activate. Or turn on and initiate an immune response. But the dendritic cells are special cells that are able to do that. In the inner core of a lymph node, you're going to find both types of lymphocytes. You wouldn't be able to tell them apart unless you gave special staining to them. You will also find macrophages. And they will all be arranged in kind of a cord-like fashion to cherry pick the bad stuff coming by. As we noted earlier, we have more afferent vessels than efferent vessels, and this is to slow the drainage through our lymph nodes. The afferent vessels will conduct lymph to the subcapsular sinus first. So we have a loose capsule on it. And so the afferents are bringing lymph in, and they're going to kind of make it bulge out or billow on out as they bring that in of our lymph node. And then lymph will then slowly percolate through the efferent, to the efferents. Located at the hilus or hilum. The slower drainage will allow for removal of unwanted cells and material. Some other important lymphoid organs. They're called lymphoid organs because they have a bunch of lymphocytes in them and held together by that reticular fiber that I talked about earlier. The spleen is the next lymphoid organ that we're going to talk about. It is our largest lymphoid organ. It is just under your diaphragm to the left behind your stomach. So your stomach, your liver's over here on the right, and then your stomach is more to the left, and right behind that is the spleen. The spleen is going to be a filter for vascular debris because lymph is coming from interstitial spaces, correct? Now we have possibility of infection in our bloodstream, correct? Do we want to kind of filter that stuff out? And so that's what the spleen is for. The spleen is a direct connection to our bloodstream to take out the things that are in our blood that shouldn't be there. So they can be dead cells, defective red blood cells. Remember, red blood cells just have a 120-day lifespan, so they pop. We need to start taking them out. And also pathogens that are present in our bloodstream. Because the spleen has some similarity in its structure, we are going to remember it's a lymphoid organ, so lymphocyte proliferation will occur after activation. So that's why in the notes it says immune surveillance and response. So some bad stuff gets into the spleen. It comes into contact with our macrophages, our dendritic cells, and our lymphocytes. That will activate them. They'll turn on, and then they'll proliferate or have mitosis occurring, and then actually start to do their job for us. Surveillance and response. The spleen, as we've mentioned in previous chapter, is a recycling place for broken down red blood cells. It releases the breakdown products from it to the liver for further salvaging. And the spleen is a great place for stored iron. So as we break down the red blood cells, we have additional iron that's being stored in the spleen. The spleen is also an excellent reservoir for red blood cells and platelets, and so if we're having some sort of actual hemorrhage occurring, the spleen will actually give you a little auto transfusion and squirt a little bit of extra blood and platelets into your bloodstream to try and save you. It's just one of the nice things that the spleen does for us.
So when the spleen is removed, is that why there tends to be like higher platelet levels, higher those types of things in the body just to breathe because there is no spleen?
When the spleen's removed, will you have higher platelet levels, things along that line? I actually do not know about the answer to that one for sure. But one of the things that happens because if you have an accident and have your spleen removed, because it has the thinnest capsule of all of those organs. So it can rupture quite easily, and when it ruptures, because it's plugged into your arterial system as well as your venous system, that's what causes you to potentially hemorrhage, and that's why you have to remove the spleen when that occurs. So you remove a very important filter. So you will actually have more of what I call the defective cells in your bloodstream a little longer than you would, popped red blood cells and stuff like that, than you would if you had a spleen. So it's taking out the bad cells. And so now the liver is going to have to pick up that job for you. And also has a tendency to actually be very helpful in removing pneumococcal pneumonia from your blood. And so people who have a splenectomy actually get the pneumococcal vaccine even at a young age because they're more at risk for pneumococcal pneumonia.
So the liver would take out extra things the spleen wouldn't?
Yes, the liver is a recycling plant also. It's just that the spleen typically is kind of plugged in to actually take care of the recycling process first and then feeds the stuff to the liver for continuation.
Okay.
And when the spleen's gone, the liver has to do it all. Anatomically, when you take a look at the spleen, we have two distinct areas, red pulp and white pulp. The red pulp sounds like blood to me, and indeed, venous sinuses, which remember are kind of big, leaky capillaries containing red blood cells, platelets, B cells, and macrophages are all in the red pulp. The white pulp they look like our follicles or our nodules, and they contain T and B lymphocytes and macrophages. So these are not areas where we find a lot of red blood cells in the white pulp. We do find lymphocytes in the white pulp. White pulp should make you think of white blood cells, and that's indeed what we have with our lymphocytes. They are white blood cells and macrophages. All right, nice picture here just showing several areas of white pulp surrounded by red pulp, and that is the area for immune surveillance and activation in those areas. The blood flow, the spleen is kind of interesting, looks in the cadaveric spleen looks a lot like a liver type of material. It's our picture right here. It also has a hilum where the blood vessels come in and out, in for splenic artery, out for splenic vein. The white pulp is where immune system activation takes place. And the blood flows from the splenic artery into the white pulp, then the red pulp, and then out the splenic vein. Splenic A for artery, then white pulp, then red pulp, and then out through the splenic vein. Thymus Our notes here say lies deep to the sternum, so it's deep to the sternum and lying on top of your heart in the superior aspect of the heart. It is considered a primary lymphoid organ. Primary means origination. Our lymphocytes are going to originate from the thymus. So primary lymphoid organ. And it is a primary lymphoid organ for T cells, not B cells.
So it's T cells only.
And the T came from thymus. So the thymus, T and B cells are both lymphocytes. And lymphocytes originate where, as far as you know? Where does all of our blood come from? Red bone marrow. And that includes white blood cells. Red blood cells, white blood cells, and platelets all originate from our red bone marrow. We call the cells from the bone marrow that are lymphocytes that do not go to the thymus for their training to become immune cells, we call those B cells. The cells that originate in the bone marrow that go to the thymus and become trained to become those type of cells, specialized cells, we call those guys T cells. You will never find a B cell in the thymus because if you go to the thymus, you are there to become a T cell. So no B cells in the thymus, only T cells. And if you don't make it to the thymus, you are going to become a B cell. So that's the distinction between B cells and T cells. So the other primary lymphoid organ is your bone marrow. So B cells, bone marrow, lymphoid organ, and then T cells, thymus, primary lymphoid organ. So we'll add that to our notes. Bone marrow, other, because there's only two primary lymphoid organs. And we get B cells from here. And remember, these are always referring to lymphocytes. It's just easier to say B cell and T cell than say B lymphocyte, T lymphocyte. And we did already mention, but all blood cells originate in bone marrow, more specifically red bone marrow. But the ones that go to the thymus and get in there become our T cells. So deep to the sternum, on top of the heart, and superior aspect of the heart. Take a look at the histology of the thymus, and let's pull this up. And to me, it looks very different from the spleen. And that's part of what you do in your histology studies. You make sure that you take a look at the picture and can figure out that, yeah, they look very different. And then from that, be able to figure out what the organ is. The spleen, for one thing I will tell you, the spleen has this white pulp scattered all throughout it. It doesn't have a cortex and medullary area. So no cortex, no medullary area, always this white pulp scattered throughout it. And if you'll notice, here is some CT right there, correct? So some CT will be found also inside the spleen, and they call that trabeculae, any sort of in-shooting of connective tissue, which we also found with a lymph node. Capsule looks very different. If you go ahead and take a look at your picture of the lymph node, capsule of the spleen looks very different than your capsule of a lymph node. If you'll notice, it's actually doesn't have a lot of spaces underneath it for subcapsular sinus. Then when you go and take a look at the thymus and realize I see no nodular follicle pattern here. So no collection that we saw with our spleen or our lymph nodes. Instead, we have light areas surrounded by dark areas. Wouldn't you say? So light areas surrounded by dark areas, and some people describe it as a jigsaw puzzle because of how they see the separation between various areas of the thymus. So they will describe it as a bit of a jigsaw pattern to the puzzle. But light areas surrounded by dark areas. So here, no follicles because there's absolutely no B cells in the thymus. The cortex is the dark stuff, dark area, is where we find our immature T cells. And then the medullary areas are where we find our mature C cells, and those are the light areas. The thymus is very important because it plays a big role in preventing autoimmunity. So that's immunity to yourself. And it's really interesting. Foreign materials are not allowed entry because the T cells are being trained to recognize you. So in the thymus, your T cells are being trained to recognize you. Because if they don't recognize you, they will get into a fight with you. And that's called autoimmunity. And so we don't want to have foreign materials allowed entry into our thymus because we don't want to suddenly think that foreign material is part of us and we will not fight it or get rid of it. So that's an important thing. And that's why we actually have what is called a blood thymus barrier.
Does it work similar to the blood-brain?
Very similar to the blood-brain barrier. Only nutrients that we want into this will actually get in there. Our thymus is exceedingly active. As soon as we are born, it is huge. If you ever see an x-ray of a newborn, and you'll see that big heart and those little bitty lungs in there, and there's something draping over their heart, and that's their thymus. It's huge when you're born because when you're born, you are thrust into a germy world, okay? And so your thymus is going, oh my gosh, what am I going to do here? And it's just going to be really, really active all the way into puberty. After puberty, it will atrophy. Atrophies, let's say, should be after puberty. So atrophies means shrivels up. So big juicy thymus in the young, and then you can tell the age of your own cats by the amount of thymic material over your cat's pericardial sac superiorly, or cephalad in your cats. So you can actually age your cat by whether or not they have much thymus. If you can find it, and the thymus looks kind of like fatty tissue still, but it's not. In the young, can still be very generous over the cat's heart superiorly, or cephalad in the cat. And the older they get, it just looks like a little bit of fatty material and not much else. So it does atrophy as we age. And this is interesting. It just turns into fibrous CT and fat. After puberty. We do have some thymic function as we age, but it declines significantly. And so the thing about preventing autoimmunity, also preventing malignant transformation. And so those things become more of a problem for us as we age. And part of that phenomenon is we are getting decreased thymic activity to help us out with taking care of these things, which should not be having malignant transformation in our system.
Is that why a lot of times they find that as a lot of the autoimmune diseases start in later teenagehood, because of thymus?
Yeah, autoimmune diseases as we turn into adolescence and later on. There's going to be some of that definitely there, but more or less it's going to be, there's been some defective thymic training. is going to be more of the problem. So there's something wrong with that individual's thymus for those autoimmune diseases. Did not get the appropriate thymic training that this is something that belongs to us, not to actually attack it. Tonsils, that's also lymphatic tissue and forms a ring around our pharynx, our nose and throat area. We have pharyngeal tonsils that we often call adenoids. We have palatine tonsils right here, close to where the soft palate droops down. Those are the ones that you can look back in your throat and say your tonsils are swollen, right? Those are the palatine tonsils. And then on the base of the tongue, there's also some tonsillar material called lingual tonsils. So tonsils are in our oral pharyngeal areas and nasopharyngeal areas because they are designed to trap pathogens entering into your body via air or food. Again, we have nodules when we take a look at them with germinal centers. However, these guys are not fully encapsulated. And that's because take a look at this guy here in the middle. So that's a tonsillar tissue right there. And on the surface, what's going to be lining its surface in the exposed area? Stratified squamous epithelium. That's always how you can tell tonsillar material, because it has a partial capsule of stratified squamous epithelium. Because do you have stratified squamous epithelium in this area right here? No, we have soft tissue, and that's why they're not fully encapsulated, because they are embedded in the soft tissue. So we have a capsule that's stratified squamous epithelium, just like everything else in our oral pharyngeal cavity is stratified squamous epithelium. The other thing, too, with tonsils, they have little invaginations called tonsillar crypts. These are also lined with stratified squamous epithelium, and they enter deep into the body of the tonsil. And this is why they fail so much at taking care of you. Because I want you to realize you have a nice puffy tonsillar gland tissue, and you poke little holes in it. Those are the crypts or the invaginations, and everything's lined with epithelium. And what do we know about epithelium? It sloughs off. So now we have this tunnel down, and we're sloughing off, and we're filling up the crypt with sloughed epithelial tissue. And that's not bad as long as you don't have a little bacterial colony growing in the base of that. You guys follow me for that? If you have a little bacterial colony that got in there, and now we're sloughing off and we're plugging it in there, you're just creating a nice area for the bacteria to live and take over and give you the lovely cases of tonsillitis and things along that line. So their construction is unfortunate because of the normal sloughing that you get from your surface epithelium. It blocks things in there and won't allow them out. And if you have kind of chronically infected tissue, then just put a plug in it, and now the chronically infected tissue can blossom, so to speak. The other thing, too, is there are some people that this epithelial tissue that's sloughing off and filling these little crypts. So there are some people out there that actually can look back there and they see these little look like corn kernels on their tonsils, and that's actually just a bunch of epithelium, and they'll flick them off with stuff. So yeah, tonsils are kind of funky type of tissue. So the idea was good, but the reality is not necessarily so good for some people, because if you do have chronically infected tonsils, the only way to get them taken care of is to take them out, a tonsillectomy. Okay, but not everybody has chronically infected tonsils, so that means their tonsils are doing their job properly, and as we age, I'll let you know, those things wither and dry up, and you know, you'll look into somebody that's kind of aged, their throat, and you won't see any tonsillar material, and they say, oh, you had a tonsillectomy. You go, no, I didn't. And indeed, they didn't. It just starts to atrophy with time and with age. Our palatine tonsils, remember, those are the big ones on either side of the oral cavity at the end, and these are largest and most commonly removed. Because of chronic infection. Lingual tonsils at the base of the tongue, you only see it if somebody's tongue is out for examination. And then pharyngeal tonsils are also referred to as adenoids, posterior wall of the nasopharynx, and they can cause some significant symptoms. And it's an auditory symptom. When they sleep. Snoring, absolutely. And they can actually, even in the young, be these palatine and pharyngeal tonsils or adenoids can be so large that they are actually obstructive and can cause obstructive sleep apnea for the young. And so are also removed often when the palatine tonsils are removed if the parents report that the kid does a lot of snoring, because it's actually interfering with a significant restorative sleep for individuals. So, now Peyer's patches. That is also given a new name, MALT, M-A-L-T. But we're going to stick with Peyer's patches. But you will see in the notes something about MALT. Don't worry about it. Peyer's patches are lymphoid follicles structurally similar to tonsils. In other words, a dark outer area when you take a look at them histology, and a light germinal center area where you're going to find your B cells. And they are found in our ileum. Do you guys know what the ileum is? It's our small intestine. It's the end of our small intestine. So they're found in the ileum, and more, the highest concentration will be in what I call the distal ileum, or towards the end of the ileum. Do you know what the ileum enters into? The large intestine, or our colon. Now, what's in our colon? I call it poop. It's poopy material, right? Fecal material, yes, poopy material. And what lives in feces? Bacteria. So what's important is once the food gets into the stomach, we are going to pour in all kinds of things to enzymes and acid and all kinds of stuff. And that's going to kill lots of stuff, right? That's going to kill lots of bacteria or pathogens that are coming in that way. And your food stuff, as it goes through the small intestine, is pretty much clean. It's been cleaned out nicely. But once you get to the end of the small intestine and you're coming up to an area of the large intestine called the cecum, you T-bone into that cecum, and there's a sphincter there. But do you think that sphincter always works in one direction like it's supposed to? No. And so you have these Peyer's patches in your small intestine to take care of backwash. You know, they're there for backwash of the nasty stuff that's going to get into your small intestine. And when I say backwash, not the fluid, I'm talking about the bugs, the infectious agents in fecal material. And so that's what we see over here in these pictures. We see the nodules, They're a little elongated in that picture, but that's there to help us not have to deal with fecal contamination in that area. Because your large intestine has an ascending, transverse, descending, sigmoid, and rectum and anal canal area. So we'll do this. A nice big sigmoid, cecum, and that's where our appendix hangs off of. But our ileum literally does T-bone into this. And we have in this area an ileocecal valve, is what they call it, but it doesn't function beautifully like our semilunars do. I prefer sphincter. And so it opens and closes to allow small intestine material to enter into the large intestine, and that's chronically cultivated with our microbiome and bacteria. So we have to protect ourselves from that. So that's the lymphatic system. So vessels is going to be on our exam on Thursday and Friday, but lymph nodes and all that stuff, not. That will be on our next exam. And this material, this lymphatic system, is very important because it's an important part of our immune system. Because why do your lymph nodes swell? Something's wrong, and typically it's infection. If it's painful, if your lymph nodes swell and it's not painful, you guys know what the problem for that is? That's what happens with cancer. Cancer is not painful. If you have painful swelling, that's the only good thing about painful swelling of lymph nodes, is that it's an infectious process, because cancerous swelling of lymph nodes is not painful itself. Yes?
So the exam stops at the second removing month?
The exam stops. Yep. Yeah. Okay. We'll just briefly get into the immune system, because I think you guys are just about done for the day.
What is it, that glassy look?
Yeah. The glassy look is giving it away. Well, the immune system, you guys know, is really important because a good immune system keeps you well right now. And a not so good immune system allows you to be sick and coughing right now. Perfect cue for me. Thank you very much. And so what's important is that we have two branches of our well-being or protection that we need to make sure we can discriminate between. And that's why we have in the beginning of this chapter, we have this innate and adaptive. Innate is something we are born with. It is part of our construction. It's how we are made. It protects us. Our construction is protective against the nasties in our world. So that's the innate, and that's why it's called nonspecific defense, because it doesn't matter what the thing is. This was here as a barrier to prevent anything from breaking through. Well, once it breaks through, however, we need to have a system that will take care of it if it actually then passes over a few other barriers that we have. And that's what our adaptive or specific system is, and that's what real immunity is. When you get an immune response, you are telling me that you have had your barriers breached, the organism got in and activated your immune system, which has everything to do with our lymphoid tissues. Because our lymphoid tissues are full of T cells and B cells, except for the thymus, just T cells. But they're full of T cells and B cells, which are responsible for your immune system. So T cells and B cells are the big players of our white blood cells that take care of you after certain layers have been assaulted and breached. So the innate or nonspecific defense, part of your construction. And let's go ahead and let me ask you, fever. That's right, it's innate, it's nonspecific, because you can get a fever from all different kinds of infections, correct? Viral infections, bacterial infections, not usually fungal infections, but it depends where the fungal infection is, but you can get fever from all kinds of pathogens entering in the system. So this is your body's mechanism to try and get rid of that pathogen to where it does not hurt you or harm you. So fever is a beautiful example of a non-specific response to an insult. However, with our adaptive system, for example, we're having a great season with this. Yeah. So influenza, by the way, gives you fever. So fever is also something that our body is trying to keep it from furthering its quest, so to speak. So fever is an innate defense mechanism against influenza, trying to get rid of it before it actually multiplies and spreads. But when that fails, when that system fails, we will employ our immune system to take care of us. And if we want to have a head start on using our immune system to take care of us, we will go to a healthcare provider and get a flu shot, which is a vaccine, which is what I call an early warning system. So vaccines are early warning systems. You take a vaccine so that you can introduce it to your T cells and B cells, and they will then suddenly go, oh, well, let's go ahead and get ready for the big war party that we're going to have. And then when the real guy shows up, it's not like, it's kind of like those surprise parties. You know, here's the culprit coming in, trying to sneak into your immune system and attack you, and people are acting like, surprise, we're going to kill you. You're not going to make it very far. So influenza, that's a flu shot, right? Flu shot, which is a vaccine or an immunization.
So my husband and his dad has never been able to get chickenpox. So they just have an innate, non-specific defense.
No. They had an occult episode. If somebody never gets the chickenpox and they're exposed and exposed and exposed and never gets it, you should have their blood drawn to see if they actually have a titer, an antibody titer for that, because I guarantee they do, because they had an infection that was called occult. Do you guys know what the term occult means? Hidden. You're not supposed to see it. Right? Occult means it's hidden. So they had an infection that was not realized because it was hidden. In other words, their system really took care of it really well, or they had a small exposure, which also makes it easier to take care of things, right? Taking care of bad things when it's small is so much easier than when they're larger. So they developed an immunity to the chickenpox, and so whenever they get exposed, they never get it. Yeah. But that's what the immune system does. The immune system, and that's why we give vaccines to our kids. Vaccines are taking the worst parts that we hope to get a good immune response to, which is a recognition response when we give somebody a vaccine, and the system has time to build its defenses. And so then when you get exposed to it in later life, there's a whole bunch of people ready to join the party, and we're going to kick you out. You're just not going to have a chance here in our system. That's what vaccines are for. And so there's a lot of people that are anti-vacciners, but they don't understand that the vaccine is not harmful to their kids. It helps their kids' immune system. Because I will tell you all right now, all of you here have anywhere from 2 to 17 different viruses in your system at this moment. So your system is handling it. You may not feel great with 17 different viruses going on in your system at this one time, but your system can handle, it's been checked, 2 to 17 different viruses in your system at any one time, and your immune system is taking care of you and making sure you're still around. That's why you guys are actually here. Your immune system guaranteed you being here, because our innate or nonspecific defenses are there as a construction model to keep things from invading you or entering you. But once they do and overcome those various things, you need something else to take care of you. Otherwise, you will succumb. And that's an important thing. So over here, what's nice, we have that innate branch here, and it shows skin and mucosal membranes. Well, that's surface barriers, right? And then we're going to look at other internal non-specific defenses. In other words, these cells will do the same thing no matter what kind of pathogen you are. And those are going to be our macrophages. You know, macrophages are just phagocytic cells. You know, it doesn't look like you belong here. I'm going to ingest you, and I'm going to make a phagolysosome, and I'm going to digest you, and you're gone. Chemicals. We're going to talk about interferon. You may have heard of interferon before. We're going to talk about it. It's non-specific for viruses. And we'll also talk about the complement system, which is non-specific for bacteria. And then we also have inflammation, which fever is a part of, which is our body's other methods to try and get rid of this before it actually flourishes in your system. If those systems fail, then you have your adaptive immunity. You've had time for your immune system to start to recognize the pathogen and to mount a defense. And we have the T lymphocytes, or the T cells, and we have the B lymphocytes, or B cells. B lymphocytes are called humoral because these are the guys that release antibodies into your bloodstream. Humoral, antibodies into bloodstream. Those are what B cells do. T cells are cell-mediated immunity, and those are the ones that I call the Marines. They're hand-to-hand combat. They'll jump on you, and beat you up. So that's their job. So we'll get into more the specifics of this, but this we'll cover this next week, and it will tell you about whether or not you should get a flu shot. That's your decision.